Thank you for coming to our presentation today. My name's Chris Hopkins. I am the CSO at NemaMetrix.
And I'm Kathryn McCormick, and I'm the Director of R&D for NemaMetrix.
And together, we're going to tell you about our platforms for connecting genes to behavior. We're setting out to tackle the big problem of gene variation. We're going to talk about two approaches to setting up animal model phenotype, and then go through our approaches to high throughput acquisition of phenotypic data. And then finally, we'll go through some case examples where we get insight into gene function. To understand the nature of the problem, we must understand the landscape. There are about 1/3 of the genome is estimated to be involved in human diseases. This leaves us with about 7,000 human disease genes to look at. We have 5 case examples here in various neurobiology disorders, those of psychiatry, epilepsy, autism, muscular dystrophy, and cancer. We can study the function of human diseases through orthology. We can look at homologs in C. elegans and study human diseases in C. elegans animal to help us gain better understanding of human diseases. To understand the nature of the problem, though, we must understand the size of the problem. We show you here in red a diagram of the size of the variant problem. So what is the variant problem? In the diversity for each of those genes, those are the numbers of variants that occur per each gene. These are the changes that are missense mutations or stop codons. If you look at those numbers and do the average of them, you get an average of about 620 variants per gene. Looking at this variant biology will allow us to connect pathogenic behaviors to the ability to have a disease outcome. Another way to look at this is to look at it on an individual basis. If we look at any one individual in this room and ask how many variants do you have in your genome, you'll have about 500 variants in your coding regions. If we then ask how many of those genes that are causative to human disease, it occurs that about 100 of those variants are occurring in the 5% or so that are the 20% that are occurring in your human disease genes. Those 100 variants, we know that about 7% of those are causative for pathogenicity. These are the missense mutations and the frame shifts that lead to early truncated protein. What about the remaining 93%? What do we know about those? Well, currently not too much. These are the variants that are either variants of uncertain significance, that's the thus alleles, or they're variants that just have not had assignment for them yet. What there is a big need for is there's a need for functional data that will help us determine whether a particular variant is pathogenic or not.
So as Chris was explaining, the problem of variants of uncertain significance can be quite large if you look at it with just these raw numbers. So if we take an average of 620 variants per gene and 7,000 genes in the human genome that are disease genes and multiply that out, we get about 4.3 million different variants that need to be vetted for whether they're pathogenic or benign or they're still uncertain. When we think about those numbers and how we're going to measure and model 4.3 billion different mutations in the animal model organisms that we typically work with, it makes sense to look at lower model organisms. So we all know that mice are the gold standard in medical research and for modeling human biology. But we also know that mice are very expensive and take a long time to create, probably years, 2 to 3 years, to get a study completed with a mouse model organism. And they have a high degree of homology, 98% in the genetic background. However, if you look at the other end of the spectrum with C. elegans, you have about 41% homology to human genes, and you have a much decreased cost per experiment, and the time scale is very fast, so you can actually get answers in 2 to 3 weeks with C. elegans, from the creation of your transgenic line to the phenotyping of that transgenic line. So we find that C. elegans is the right level for us when weighing relevance to humans and cost per data point. So using C. elegans, we can have a whole system that assays variants of uncertain significance, and it's very quick and affordable to create the mutants that we're interested in. And it's quick to get the phenotypic readout from those genetic variants as well. So I told you a few slides ago that about 40% of the genes that humans have orthologs and C. elegans. But if we look just at disease genes, those numbers are actually enriched. And this is because a lot of the disease genes are in evolutionarily old signaling pathways. So for just looking at those disease genes, there's 80% of those human genes that have homologs in C. elegans. And in C. elegans, it's also easy to apply a lot of modern genetic techniques, such as CRISPR and MOSCI, and a lot of site-directed mutagenesis techniques. And then we find that when we apply these genetic techniques to C. elegans, the data that we get occurs in tissues and organs rather than in isolated cell lines or artificial environments. So, just a little bit more about C. elegans. C. elegans has 959 somatic cells and 302 neuronal cells. And using this very simplified model organism, we actually have the ability to assess all those different cell types and look at how the nervous system of C. elegans creates the behavior that it displays. And this makes it very good for looking at neurodegenerative diseases, but C. elegans also has NIH support for studying the genetic control of aging, and it makes a good model for cardiac disease as well, because many of the calcium channels that create human heartbeats are expressed in the muscular organ known as the pharynx inside C. elegans. And so I'm going to show you a little bit of data today from the pharynx of C. elegans, as well as other types of phenotypes that we can get from this animal. But first I want to tell you about our approaches to genotypic recapitulation of human disease in C. elegans. So we really think of doing this in two different ways. One is if there is an ortholog of the human disease gene in C. elegans that has a conserved loci, we can create a pathogenic allele that is the same point mutation that you would have in the human gene. And so we call this native locus humanization, where we take a pathogenic allele and install the same mutation at that site. The other way that we can do it is by swapping out the gene inside C. elegans with the human gene. So we take a canonical human sequence and replace the C. elegans gene, and then we have an in vivo system from C. elegans that is expressing the human gene. And we'll show you some data later. Chris has a lot of great data about how these installs of human genes actually function inside C. elegans, and they rescue phenotypically the knockouts of C. elegans genes. And with those genes, we can then add on another layer where we take a patient allele that is pathogenic, and we can install it inside the human gene that's now expressed inside C. elegans and see the pathogenicity. And this is another approach that's very efficient for genetic manipulation that we're actually very excited about. So inside particular genes, and we're here showing STXBP1, which is an epilepsy gene, there are hotspots of mutation, and inside these hotspots, lots of different allelic variations occur. And so in a single injection for C. elegans, we can create multiple HR templates that recapitulate the changes to the human genome. And so we have the same guides. We'll put the guides on either side of the hotspot, cut there, and then the recombination can occur with any one of the HR templates that we put in our mix. So then from a single injection, you could get 20 or 30 different types of mutants inside that same hotspot inside the gene. So it's a very efficient way to get a lot of different allelic information. And for those of you who use cell culture normally, you may be wondering, can I integrate this into my workflow? Is it even possible for me? And the answer is yes. We provide our animal models as kits, so if you want to get a particular gene modified, you can get it modified, and then we will send it to you in a format where you can just pour it onto a plate, incubate it at room temperature, and then 3 days later get the phenotypic information from a microfluidic format. So data acquisition is easy, and animal acquisition is also easy. So now I'm going to go in a little bit deeper and tell you about the phenotypic approaches that we take with C. elegans. This pictured here is the NemaMetrix screen ship system, and this is a microfluidic system that works kind of like a flow cytometer. So we flow in a worm to a particular position, we take a lot of measurements from that worm, and then we flow the worm out and get the next worm in. And so it works a little bit like a high content analysis system that some of you might be using in your cell types. And with it, we can assess a lot of different important phenotypes that are both morphological and behavioral for the worms. So this includes growth, movement, neuromuscular phenotypes, fertility, digestion, stress levels, and more. And so I'm just going to show you a little bit example of our growth module. So with this, we have a C. elegans trapped inside a microfluidic channel, and that's what you see at the top. And then we have the software is automatically determining the length and volume of that worm because the worm is in a channel of known dimensions. And so with this, we're able to get not just this worm's specific measurements, but also a lot of data from different worms, and we can look at population averages for length, for volume, and do statistics on that. So many of the mutations that we've looked at actually cause worms to be delayed in their growth. And we can see that really easily with our software. We also have integrated payback. So I was telling you a little bit about the neuromuscular phenotypes. And those phenotypes, those waveforms from the neuromuscular pump called the pharynx can be seen here in the middle panel with all the yellow, blue, and green dots. And so you might see that that looks a lot like a heartbeat. It has an upward going peak, it has a downward going peak, there's a duration, there's an amplitude, there's a lot of detailed information inside the pharyngeal measurement that we take. And it's a good readout of neuromuscular function because there's a known number of cell types that create that pump, and there's a known number of muscles that have to contract in a coordinated manner to also create that waveform. So we measure the waveform, and we simultaneously take a video of the worm. And on the top you can see the worm itself. It's pumping away, it's moving, it's got its head wiggling, its tail's flipping around, and at the same time we're looking at the center line of that worm and how much it's moving, and we have a degree of information about its health from all these phenotypes. So in the vein of saying that this animal is as easy to use as cell culture, we can also apply stains, dyes, and fluorescent tagging here. So I'm showing in this slide lysosomes tagged with our worm dye lyso, and this looks at apoptotic corpses and lysosomes inside the animal. And so you can flow the worm into place, take a measurement of the lysosomes that are present, and then move on to the next worm. And here's some images that our customers have taken of other fluorescent tag proteins that we have created. So on the left side, we have nuclear labeling inside the gonad of C. elegans. So this gives really detailed information about fertility and how cell cycle division is going along during the creation of new embryos. And on the right-hand side, we've tagged mitochondria. And we're looking here at a biosensor strain for mitochondria. And so these are just two examples of the types of genetic tagging that we can provide that gives you mechanistic information of organelles. So with that, I'm going to give the microphone back over to Chris to talk about how you would apply these in clinical and other settings to look at how genes create human disease behaviors.
Okay, one of the first clinical examples we have is Elia Di Schiavi. He's from the Italian Institute of Biosciences and Bioresources. Elia was interested in the DAT1 gene. This is the dopamine transporter, and he wanted to understand its role in Parkinson and other neurodegenerative disorders. The DAT1 gene, it acts presynaptically. When you get a nervous impulse comes down, the synaptic vesicles fuse and release dopamine into the synaptic cleft, and that dopamine then acts on the downstream receptors and transmits the signal downstream. Well, then the DAT1 transporter then takes that dopamine back up and brings it back into the presynaptic side, where it's used and recycled and refilled into vesicles and being ready for the next electrical impulse. To study this gene, Elia took use of a knockout of the DAT1 gene and asked what was that effect on function. For the functional assay, he used the oxydopamine assay. In the oxydopamine assay, oxydopamine, when it is brought into the cell, becomes toxic and leads to neuronal cell death. So what he did is he monitored the dopaminergic neurons for the degeneration. And when we look at wild type and look at when it's exposed to oxydopamine, ask what is its levels of reduced or loss of degeneration of neurons, we see that about 50% of the neurons have been degenerated. When you have the knockout and you've lost the transporter function, you see a significant phenotype where these animals become highly resistant to neurodegeneration due to oxydopamine exposure. Next, Elia wanted to go look at see if he could rescue it with human transgene expression. So he took the human cDNA for the human DAT1 transporter and put it into the worms and asked, could it suddenly restore the sensitivity to oxydopamine? And he saw a significant level of restoration of sensitivity. And finally, Elia wanted to look at two different types of mutants that are known to be pathogenic in humans. One of these is sort of an obvious mutant. It's a frame shift mutant that occurs early on and should wipe out function, and it does. That's the first bar in red. But then he looked at another variant, one that's perhaps a little more surprising, an alanine to valine change, a subtle change, but it's known to be pathogenic. And he saw in this same system that it also totally lost transporter function. In another example, we have Kathryn McCormick. She's a pediatrician at Harvard. She was interested in trying to understand the sodium leak channels and their role in neurotransmission. Sodium leak channels act to control the tone of the membrane, and so they control the strength of the signal. And when they're defective, you lead to altered neurotransmission. She wanted to look. She took the advantage of the C. elegans that has two different alleles in it, known to be one severe and one is a mild allele. When she looked at those in an assay, she saw a differential response. The assay she used was the aldocarb sensitivity assay. When aldocarb is exposed to the animals, they get paralyzed. If you expose it at a low dose, you get a slight paralysis. That's what we see with wild type, about a 25% paralysis. When she looked at the mild allele comparatively, she saw that it didn't seem to have much difference in effect compared to wild type. It was also showing only a slight paralysis. Yet when she looked at the severe allele, she saw a fourfold boost in the level of paralysis, indicating a strong measure of that phenotypic output. Next, she wanted to look at a patient allele. She had a patient that had a defect in the nalcine gene. And so this models the nalcine gene, which is involved in hypotonia. The NAC1 gene is a homolog for the nalcine gene. And this patient had a mutation, but it was a variant of uncertain significance. And what she was able to do by using CRISPR to install this into the native locus of C. elegans, she was able to create an animal that expressed this mutation and asked, is it pathogenic or not? And when she did that, she saw that it phenocopied the severe allele, and she had an ability to sort of diagnose her the disease and have a target for further therapeutic development. Another example, we have Ralph Grenier. Ralph is the director of the Seattle Autism Center. Ralph was interested in the CHD8 gene and its role in autism. CHD8 gene is a helicase, and I show you here sort of to remind you of what this most consider the classic example of how a helicase works, and it works in DNA replication repair to help unwind DNA and for replication. But helicases are also involved in chromatin remodeling, and that's the case with this gene where it acts in neurodevelopment. Shown here is a scheme of the various mutations that are seen in patients. These are the red lollipops above the bar, and if we zoom in into a small region, we can see that there are 4 red letters there that represent pathogenic variants. We are going to be using a shotgun approach to install these variants, but we're also going to be installing a lot more variants. The rest of the variants have unknown significance in this region. So in a shotgun approach, we're going to be making all of these variants and assessing that for whether or not they lead to loss of function. In one of our own examples with our own team, we have Tom Marshall. Tom was interested in trying to understand the synaptic binding protein number 1. This is the UNC18 homolog. It works in synaptic vesicle release, binding to the snares, leading to coordinating the release of synaptic vesicles. When you are defective in this gene, you lead to loss of coordinated vesicle release. Tom used this, the UNC18 homolog, as a site to engineer in point mutations of clinical importance, and he chose 5 mutations to put in. These are shown on the, there's the S42P mutation, showing when installed in the animals and tested under what we call the worm race assay, where you put down worms on one side of a plate and let them have a timed race to food. He saw that those had reduced amount of ability to get to the food relative to wild type. In another allele, the knockout allele, the R38X allele, he saw that it was severely hampered in the worm race assay. 2 other alleles were looked at, the R39H, the R292H and the R406H allele. Both of these are flagged as pathogenic, but they don't appear to be as pathogenic as the other 2 alleles. And finally, he looked at another allele, the proline to leucine change, and saw that it didn't appear to have much effect in this particular assay. This allowed us to take and sort of score these pathogenic alleles for whether or not they had pathogenicity, and we saw consistent with what other people have annotated that the 4 pathogenic alleles are pathogenic. And what was interesting is that the variant allele that was a VUS allele in our system is being reported, and we gave it an assignment of assessing it as a benign allele. Tom wanted to ask, is this really relevant to humans? And one way to establish that relevancy is with the complementation test. So Tom took the cDNA of the human syntaxin binding protein and inserted it into the animal. And then when he looked at that cDNA compared to the null animal, he saw that cDNA was capable of completely restoring pumping frequency as measured on this screen chip. And in another parameter that is measured with the screen chip, which is the pumping duration, the knockout allele shows a 2-fold increase in the pumping duration, but that was completely rescued with the syntaxin binding protein. Finally, as the last slide on the applications, we have Jaegil Shim at the Korean National Center, Cancer Center. He was interested in the EFGR gene and wanted to understand its role in cancer biology. And to study it in the C. elegans system, to humanize it, he made a chimera where he took the TK domain coming from the human EGFR receptor and fused it to the cytoplasmic domain, the extracellular domain of the LET23 gene. And then on top of that, he built in various pathogenic mutations, and one of them being the leucine to arginine mutation. When he looked at this leucine to arginine chimera and tested it for a phenotype that is strong indicator of a carcinogenic output, which is this MUV assay, he saw a significant amount of MUV formation. But when he exposed it to various chemicals, he found that one chemical was leading to a reduction in the amount of that cancerous readout. And so, what this implications of this is that the humanizing C. elegans and then these fast phenotyping assays become a platform for helping us find therapeutics that reverse pathogenic phenotypes that are specific to patients, so a form of personalized medicine. To summarize on this, our genes to behavior system, we have developed functional assays to aid us in understanding variant biology. We do this in the whole animal models that allows us to do this in a whole animal model context. Because we need millions of animal models to model the variant space, we choose to use C. elegans where it's very fast and affordable to knock in human mutations. And I'd like to thank you for your time and attendance.